This might work better now. Okay, we'll see. Okay, so we'll keep trying to post the videos. No guarantees for today. If it doesn't work, then maybe I might try to ask Sean to bring an actual video camera. So we'll see how this goes. Okay, so as I said, the lectures so far kind of reflect the size of the field, right? So we've gone past the first unit, which is generally about methods and how we come to know what we know. Now it's about what we actually know. And on the syllabus, it's sort of nicely laid out. There's 1 day on vision, 1 day on audition, 1 day on the chemical census. But that's not actually how big the fields are, right? So it turns out that in cognitive neuroscience, there's a ton of people who study vision, a ton of people who study audition, and like a few people who study chemical census. And so the actual length of the lectures will reflect that, right? So today I'll talk about audition, and that's going to go into about half of the next lecture. The next lecture I'll cover chemical census. Okay, so the goal is to talk about audition. Now, if you get very, very interested in this, I should say that I have an advanced course, Advanced Research Methods in Auditory Cognitive Neuroscience, that actually meets right after this one. And so if you find this stuff particularly interesting, you could feel free to enroll in that next semester. So we'll start by talking about what the sound stimulus is, right? So what exactly the physical changes are, a physical signal is in the world that we're trying to detect. And then we'll move into talk about the whole auditory pathway, right, from the ear, right, from the outer ear through the middle, inner ear, and then all the way up to the brain. And then we'll talk about some higher order auditory functions such as speech and music. And my area of research is music and the brain, so I'll talk a little bit about that as well. So some neuropsychological populations that have some auditory or central auditory or peripheral auditory disorders. And so what exactly is sound stimulus? So we know that we're walking around in a medium of air, right? So fish are swimming around in a medium of water. But to the first approximation, all sound stimuli are vibrations of whatever's in the medium, right? So for us, that's air. For a fish, that's water molecules. But for us, when we're walking around in the world of sound, we have stimuli that are vibrating, and that causes a propagation of air molecules that are being sent around in the medium, right? So in air. And so when these sound waves get sent around, that causes the compression, right, and rarefaction, or just back and forth movement of air molecules that are predictable in certain ways, right? So that's just one example of how air pressure might be modulated by sound as the sound is emitted by a source such as a tuning fork, right? So how many of you have seen a tuning fork? Right, okay, so that's just a simple metallic piece of two tines, right, that you hit, and you add some energy to it, and that thing then vibrates with in a very periodic manner, then modulating the air pressure, which then goes into your ear. Right, so when people are talking, you're just modulating the air pressure that's right outside your mouth, and that then through the airwaves gets into your ear. Right, so sound is a modulation of air pressure, and so by the compression and rarefaction of air molecules, that then hits your eardrum, right, which then receives the sound and then funnels it into your auditory system. Right, so sound stimuli are changes of air pressure over time, and because we can describe these changes of air pressure in very periodic ways, that is then susceptible or analyzable using frequency domain analyses. And this is a slide that was from previous lectures where we talked about the Fourier transform of the EEG. Right? So this is Joseph Fourier, this French mathematician, who famously discovered that any complex time series, right, such as EEGs or such as sounds, can be broken down into a series of sinusoids with different frequencies. So all that this means is by doing a bunch of calculus, like what you won't have to do here, you can actually take any complex signals such as sounds or such as brainwaves and analyze them and just describe them as a series of sinusoidal back and forths. Right? And so that then gives you an idea of the frequencies, like multiple frequencies that are in the energy that is in the sound, right? Just like they're in the brain. So you can describe, for instance, this example of a complex signal as some high frequency components and some medium frequency components and some low frequency components. And when you do that, that's very helpful because then you can visualize it differently. And so now this is a time domain and frequency domain waveform of a speech. Actually, this is a piece of music that is recorded, and you can visualize it as a sequence of different frequencies over time. Right? So on the y-axis, I'm showing frequency. On the x-axis, I'm showing time. And on the color, right, which is the third dimension in a way, I'm showing that the brightness corresponds to amplitudes. So the sound stimulus can be analyzed as a function of time-dependent frequency modulations of different amplitudes. Okay, so these are some physical descriptions of the sounds themselves. Now let's talk about how we receive them in the ear. All right, so before we talk about the inner ear, we'll just have to talk about the ear. Okay, so the ear, everybody has one, right? So some of you probably know some people who are hard of hearing, right? So people who are hard of hearing nevertheless have an intact outer ear, usually, right? So the outer ear is just the pinna, which is the funnel-shaped, conch on the side of your head. And that the job of that is just to funnel sounds into your auditory system. And so that goes through then the auditory canal, which then hits the eardrum. Right? And so the vibration of the eardrum is what translates the signal into the auditory system. And so between the level of the eardrum and the level of the round window, that's generally known as the middle ear. The resolution is a little off. So that's the middle ear. So that's just the period, the area between the eardrum and the round window. And what's really important in the middle ear are the little ossicles, right? The three little middle ear bones that detect the vibrations from the level of the eardrum and then transmit them towards the inner ear. And so from the level of the round window on, you're looking at the inner ear. And so I do want to focus a lot more on the inner ear. So as I said, it starts with the round window, and then there's the most important organ of the peripheral auditory system, which is the cochlea, right? And there's also some additional organs within the system. That's the semicircular canals, which are right here, which are important for your vestibular system, and which is useful for detecting your positioning within the world. So we'll talk about the vestibular system the next day, but today we'll focus on the cochlea and how it talks to through the auditory nerve to the rest of your auditory system. So the cochlea is about the shape, the size of those little erasers at the tip of your pencils. Right? So it's a small snail-like structure, and so this is a microscope image of the cochlea, and so it's like a rolled-up snail. Right? And so if you take this this canal of the cochlea and you take a cross section of it, this is what the cross section looks like. It's got a bunch of different fluid-filled channels, and the fluid-filled channels are better illustrated here. We have the cerebrospinal fluid that's kind of throughout the whole area, but you have importantly two major membranes that are interacting with each other. The top one is called the tectorial membrane, T for tectorial. The bottom one is called the basilar membrane, B for bottom, which is for basilar. So the interaction between the tectorial and the basilar membrane is what gives rise to the neurotransduction for sound. Am I in your way? Okay, yes. You're not used to actually having a person standing here. It's a bit surreal. Okay, so the interaction between the tectorial and the basilar membrane is what gives rise to the transduction of sound. So how do those two work together, right? And what's kind of moderating that relationship? Well, it turns out that this whole thing, it's called the organ of Corti. I think that's, yes, organ or Corti. It should be called organ of Corti. I don't know why there's a typo on that picture. So if you zoom in on that organ of Corti, which includes the tectorial membrane, the basilar membrane, and all the cells in between, you'll see that the cells that are doing all the work here are all here. They're called hair cells. And there's two kinds of them. There's one row of inner hair cells, that's the first kind. And there are 3 rows of outer hair cells, that's the other kind. And it's this back and forth movement between the tectorial membrane and the basilar membrane here, that opens up the inner hair cells, which then translates the mechanical movement of the vibrations within the whole system into electrical potentials that are action potentials, that is the currency of the whole system, of the whole nervous system. Right? So again, this is another nice picture of it. Right? So the tectorial membrane is moving against the basilar membrane, and so when the tectorial membrane opens up, when it's activated, right, so it opens up all the channels that are tied to the hair cells, which are in the inner hair cells, and so that then opens up the hairs of the inner hair cells, which then enables the signal transduction via the chemical cascade that is typical of action potential. Okay, so what is special about the outer hair cells? So again, I've said that there's stereocilia, which is just another word for the hairs of the inner hair cells, and then there's 1 row of inner hair cells and the 3 rows of outer hair cells, which tend to be a little bit bigger, right? And when the whole organ of Corti is excited, the tectorial membrane is moving up, right? Which then opens up, kind of drags open the inner hair cells, which then enables this transduction. And so that's exciting the whole system. Now, what do the outer hair cells do in contrast? Right? So the 3 outer hair cells are kind of sitting there reacting to the 1 row of inner hair cells. But what it's doing is it's acting as a control mechanism for the whole system. And it's acting as a kind of gain control, so that when inner hair cells are overly activated, it detects that there's high level of activation in the inner hair cells, so that it damps down on the outer hair cells, right, by pulling up the basilar membrane. So again, so it's this this relative movement of the tectorial versus the basilar membrane that then controls the whole system, which enables the auditory signal transduction to happen. So what's the good of outer hair cells doing this kind of gain control thing? So raise your hand if you've ever been to a crowded party, or a crowded like rave party or party with lots of music in it. Yep, okay. And then when you walk out into the quiet night, raise your hand if you felt this ringing in your ears. Right. So that's pretty common, right? So that is actually a healthy example of your outer hair cells working. So what's happening there is when you're in a very loud environment, your outer hair cells are protecting your inner hair cells from having to transduce so much activity that it kind of overwhelms your auditory system, right? So it's protecting your outer hair cells. That's what gain control means, right? It's just letting in just enough information so that your system can tell what's happening, but not too much information so that your whole system is shot. And so when you go from a quiet environment suddenly into from a loud environment suddenly into a quiet environment, suddenly you are there's an overactivity of your outer hair cells, which is why you hear a ringing in your ears. Right? So that's in a sense the outer hair cells saying, oh, it's a very loud environment, although it isn't anymore. Right? So then there's that change, and that's what gives rise to ringing in the ears. And some people actually have abnormal ringing in their ears, where the outer hair cells kind of won't shut up even when they're supposed to, and so that's known as tinnitus. Which is overactivity in the outer hair cells, which causes ringing in your ears. That's true. Okay. So a little bit more about, so we spent a lot of time on the organ of corti, a little bit more about cochlear mechanics and how the whole cochlear works as a whole system. So as I said, the inner ear begins with the round and row, right? And then this cochlea is usually this rolled up structure that looks kind of like a snail, a small snail. But when you unroll that cochlear structure, what you can see is that there's the basilar membrane, tentorial membrane that's laid out in the middle, throughout the length of the cochlea. And what sounds are doing as they're coming in from your outer ear is that they're getting filtered through a series of the different parts of the basilar membrane that is organized by frequency. And so organized by frequency, tonotopically organized is just another word for being organized by frequency. And what that means is that one end of the system is sensitive to high frequency, whereas the other end is sensitive to low frequency and the whole gradient in between. So as the sound comes in, you can think of it as a traveling wave, right? And you can think of the whole basilar membrane as a system of filters that starts out by filtering the high frequencies, and then whatever doesn't get filtered out in the high frequency range goes eventually down to the low frequency range. So you need the whole length of the cochlea in order to have high fidelity hearing throughout the whole range of your auditory system. So this is a nice little video of a model of the cochlea. So you can see it's kind of a zoomed in version of how sounds go from the outer ear all the way to the inner ear, and then you can see how sound activates an uncoiled version of the basilar membrane. Awesome, right? So that's just a model of how the whole thing works. Now, what happens when some parts of the hair cells, or some parts of different cells along the basilar membrane, undergo degeneration, right? So one example of hearing loss is sensory neural hearing loss. Right? There are multiple kinds of hearing loss, but sensory neural is a pretty common one, and that's the most common age-related hearing loss, for example. So usually when you have a normal organ of corti, again, you have your one row of inner hair cells and your three of outer hair cells, and then they're regulating that relative movement between the tectorial and basilar membrane. But when you have injury to your hair cells, what happens is the hairs themselves get shot. And so when you have injury, like partial or severe injury, you get the cells that are normally kind of the stereocilia of the hair cells kind of get disintegrated, so that eventually, if you have a sustained injury for a long time, you actually lose the organ of corti. And this can happen if you're just getting too much exposure to very loud sounds. Right? So loudness is something that we should talk about because that's a big part of what we're exposed to in sound stimulus. So when we're talking about a loud sound, generally, physically, it means that there's more energy, right? So a sound that has a high amplitude is generally correlated with loud sounds, the psychological percept of the sound being loud. But there are lots of nonlinearities in this system, right? So So it's also completely frequency dependent, for instance. So sounds that are within this range of 1,000 to 5,000 hertz, if given the same amplitude, will sound louder to you than sounds that are outside that 1,000 to 5,000 hertz range. Okay, so there's a frequency dependence of loudness. Then the system is also sensitive to top-down modulation. Right? So that means that just because there's more energy doesn't necessarily mean that a sound is loud. Right? So you can have someone shout from a long way off, and the amount of energy that's hitting your eardrum might be small, but you might still perceive it as loud because you're used to hearing shouts as being loud. Right? So there's some psychological aspects to it as well, and so that's what we would call top-down modulation, or in other words, a kind of information that comes from your long-term experiences, not necessarily from the stimulus per se. And I should say that it's measured in log scale. So something that's 10 times louder is 1 decibel louder, and then sometimes that's 100 times louder is 2 decibels louder. Right? So it's measured by a logarithmic scale. And so this is from your textbook. Right? So the minimum audible intensity is pretty much you can't hear anything at all. So it's kind of akin to the Brownian motion of just air molecules floating around, not really going in any particular direction. And so that's so quiet that you start to hear your own ears ringing. But very quiet rooms. So we have some sound attenuated chambers in my lab that are providing a lot of attenuation from just background sounds. So this room, for instance, is not, there's a lot of background noise, from the fans and whatnot, and that is already providing more than 40 dB loudness in this environment. But spoken conversation is usually about 60 to 70 dB, and then a loud rock concert is over 100 dB, and so on. And I should say that if you're exposed to more than 100 dB for 20 minutes at a time, you can start to experience some noise-related hearing loss. So don't go to too many loud rock concerts without hearing protection. And what I meant to when I said that loudness is frequency dependent is that the same sound. Right? So this is a famous kind of plot known as equal loudness contours. And so each line here is just representing how loud a sound sounds to you given a certain amount of energy. Right? So if you're looking at the same loudness level, let's say at the 60 dB mark, it will sound much. So the lower it is, the more audible something is. So given a 60 dB mark, if the sound is within the 1,000 to 5,000 hertz range, you're much more able to hear it. It sounds much louder to you than if it's in the 20 hertz or 50 hertz range, right? Or than if it's over 10,000 hertz. Right. So that's this kind of quasi U-shaped or W-shaped equal loudness contour just shows you that how loudness depends on not just on the amount of energy, but also on the frequency of the sound. So what happens when we're getting hearing loss, right? So usually, the most common kind of hearing loss, age-related hearing loss, affects high frequency. So ski slope, people call it ski slope hearing loss. And what that ski slope refers to is the shape of the audiogram. Right? The audiogram is just a measure of how well you can hear sounds at different decibels. And so, does anyone have an audiogram done on themselves? Oh, okay. Yeah. So I've tried this on myself. So it's just a machine where you put the headphones on and you're played different sine wave sounds that are relatively quiet, right? And you just raise your hand and you can hear it. And so if you're at a 10 dB, right, so that's normal hearing. But if your mild hearing loss, moderate hearing loss, and so on, that just tells you how many dBs it requires for you to reliably hear something. Right? So if you have 40 dB hearing loss, it means that something has to be 40 dB before you can hear it. So a normal audiogram is just so you usually get two lines. For the data within the audiogram, and that just tells you how how much energy it takes for you to be able to hear something. So usually your normal hearing, normal person audiogram would have two lines that are just across the top like that. But this example, this person has slow hearing loss, which means that once we get past about 1,000 hertz, it takes a lot more energy, right? It takes a lot more decibels for them to reliably hear it. So like my grandmother had this kind of C-slope hearing loss. And so just as an example for what it actually sounds like to have high frequency hearing loss, this little clip I'm about to play, it's a speech, it's a clip of speech that starts normal, and then it gets filtered such that the high frequencies get filtered out. Right? So that's so it just simulates what hearing loss might sound like.
A typical audiogram comparing normal
and compared hearing. The dip or not at 4 kHz as shown, or at 6 kHz, is a symptom of noise-induced hearing loss.
Did you guys hear that? Not very well. Let me try something else.
A typical audiogram.
Let me try that again.
A typical audiogram comparing normal and compared hearing. The dip or not at 4 kHz as shown, or at 6 kHz, is a symptom of noise-induced hearing loss.
Right, so you could hear how it started out like a normal speech and then it got filtered out so that it sounds very muffled. That's a little bit like how a person with hearing loss would hear it. So that's why when they say like, can you speak more clearly, it's not that you're not enunciating, it's just that the high frequency components of your sound, such as the E parts of your voice get blocked out as a result of the hearing loss. So that's what high frequency hearing loss sounds like. And what could you do to protect your hearing? You can, of course, protect your ears using earplugs and over-the-ear muffs. And I should just say that combined methods of protection always work better than single methods of protection. Right? So you're attenuating a lot of different frequencies, and ideally you want all frequencies to be attenuated similarly. But that's not necessarily the case with the smaller kind of earplugs. Now, what happens if you have complete loss of cochlear function? Some people can also get cochlear implants, which is really quite a modern medical miracle. What that's doing is you can have an implant kind of through your head just by kind of putting a small hole through your head that then goes right into the cochlea. That is essentially, it's a strip of electrodes. Right? So instead of having hair cells that are doing the signal transduction, you're inserting a strip of different electrically sensitive electrodes, such that when the sounds, which also then get amplified outside your head, when the sounds come through the system, they activate the electrodes instead of the hair cells, which then activate the auditory nerve, which is coming out of the cochlea, right? So it's just a really neat way of getting the sound signals to, from the outside world to talk directly to your auditory system while bypassing your peripheral auditory system, aka your cochlea. So that's a really neat way of doing things. And so this is what it looks like on a child. And it's kind of a very active area of research, what these different configurations of electrode arrays and how they can help you hear different kinds of things. Right? So it turns out that, for instance, needing to hear speech requires kind of a different set of electrode arrays in different settings than hearing music. Right? So can we actually engineer better cochlear implants so that you can hear music as opposed to speech? Right? So that's a very active area of research. Any questions so far? Yeah. They're irreversible. Yeah. That's a really good question. So cochlear implants, so the technology has changed a lot over the years. With cochlear implants, at first it would sound like kind of filtered speech or vocoded speech. So instead of hearing kind of the full fidelity of, for instance, what I'm saying, you might hear individual formants. So you might hear it as kind of moving bins of sounds. Yeah. So you don't get very good frequency resolution. So for instance, different notes, like if you're trying to play like a violin, they have different notes very close together, different pitches, they wouldn't be heard very well with this system. Just because these electrodes, there's kind of a mechanical limitation to how close they can be, right? And there's also electrical limitation, such that when electrodes are too close together, they start interfering with each other, such that activity in one electrode kind of interferes with the electrodes next to it. So it's kind of a fine art for working on these electrodes such that you can get as much information as you need. It's still not going to work as well as a normal apparatus. Okay. Yeah, so is this normally this loud? It's like very loud fan noise. Oh. All right. So it's especially appropriate that we're talking about auditory system today. Anyway, so let's move on to talk about the auditory pathway and so there's a number of different way stations of neurons all along the auditory pathway from the periphery all the way to the brain itself, and some of them are important. They're important for different reasons, but there's a cluster of neurons specifically that are useful for sound localization. So I do want to spend a little bit of time on that. So the auditory pathway, this is kind of an overview of the whole auditory pathway. You have something much like that in your textbook, of course. And this is just showing you how the human auditory range is 20 hertz to 20,000 hertz. Right? So if you're interacting with sounds that are between the range of 20K to 20 to 20K cycles per second, that can be detected as audible sounds. If you're below that, you're starting to not perceive it as sounds. If you're above that, you just don't hear it as sound at all. So this is how your textbook portrays the different areas of the auditory system. So it goes from the cochlea, which is starting the acting potentials, which is initiating the first step of the auditory pathway, the auditory neural. And there's the auditory nerve, which comes out of the cochlea, which then synapses onto the medulla, and that is where the cochlear nucleus is. And then from the level of the medulla, it then goes up to the pons, right? And the pons houses this first crossover where information from the left ear goes into the right brain and vice versa. And so where this first crossover is, the areas are called the superior olivary complex, or superior olive. And that's a combination of two sets of neurons, the medial superior olive and the lateral superior olive. And those are super important because they without them, we wouldn't be able to localize sounds. Right? So that's how you can tell, like, I'm standing to your left or your right. Without that, you would not be able to localize sounds. So medial versus lateral superior olive. We'll talk a little bit more about how those work in a bit. But just as an overview, to continue the overview of the auditory pathway, the auditory input then goes up to the level of the lateral lemniscus and the inferior colliculus, and then medial geniculate body, and that's medial geniculate body is within the thalamus, which is the most important way station in your midbrain before information gets sent to the level of the cortex. Okay? So after the medial geniculate body, that's the cluster of neurons within your thalamus that specifically talks to the primary auditory cortex. Right? So then at the level of the primary auditory cortex, that's the level of the brain that you could image with a functional MRI and so on. But there are multiple way stations, right, which make the auditory system pretty complex, but they are also very temporally sensitive. So this is just another list of the areas that are part of the way stations of the midbrain auditory system. So that goes from the cochlear nerve through the cochlear nucleus to the super important olivary complex, which includes the medial and lateral portions that are important for sound localization. And then we have the lateral meniscus, which is important for detecting amplitude modulation, and I'll talk a little bit more about what that is as well. And then you have the inferior colliculus and the MGN, which is your last line of relay towards the primary auditory cortex. So I just want to give you a bit more of a sense for where exactly this medial and lateral superior olivary complex are sitting in your brain. So this is just a diagrammatic, and this is the actual picture of where the synapses are and where the areas are. And so it's part of your midbrain. And importantly, that's where input can come in from both left and right ears. Okay. So your left superior olivary complex is receiving input from your left as well as your right ear, and vice versa. So how do those work, right? So your medial superior olives are super important for detecting interaural time difference. So what exactly does that mean, right? So that's just the difference in time of arrival between the two ears. Right? So if I am standing to your right, I'm standing to most of your right, my voice is going to hit your right ear some microseconds before it hits your left ear, right? So the job of the medial superior olive is to compare when the sound hits your left and right ear. So that's the interaural time difference, which is ITD. In contrast, the lateral superior olive, or the LSO as opposed to NSO, is important for detecting interaural level difference. Right? So if I'm talking to your right ear is going to hear me as a little bit louder than your left ear. Right? So the job of the LSO is to detect that difference. I once played in a band called Superior Olives. It was a lot of fun. We were all auditory neuroscientists. So the MSO, so how exactly does that work? So what's in the MSO is a bank of neurons that are actually coding for, as delay lines, right? So that are coding for coincidence detection between the two ears. So right here you have a bank of neurons that are the medial superior olive together. And that, as I said, receives input from the left ear and from the right ear. Right? So if your loudspeaker is coming from the left, the sound's going to obviously reach your left ear first. And so then it goes through a series of delay lines into the different neurons that are within that pool of neurons that comprise the medial superior olive. And so similarly, the sound's going to take a little bit longer now to reach your right ear. And then you also have a set of delay lines that will be implemented that are going to activate your the different neurons that are within your bank of neurons within your medial superior olive. So what the medial superior olive can do, right, using its bank of neurons, is to detect when input comes in a coincident manner, right, at the same time from your left ear and your right ear. Right, so that's what coincidence detection means, is that this bank of neurons is detecting simultaneous input from your left and your right ear. And so this bank of neurons now can give you a little map, right, of where sounds are coming from, which is a really elegant kind of model for how this whole system works. And so this was found by, first proposed by Lloyd Jeffress, and then also been identified in a couple of different animal systems. And so 50 years since it happened, it's been identified in the barn owl and then in the cat. It's pretty cool. Okay, so we talked about the superior olive a lot. Now the next levels up, there's the lateral meniscus and the inferior colliculus. Those together are very important for detecting amplitude modulation. So what exactly is amplitude modulation? So as I said, amplitude is the level of the sound. Right? So loud sound usually has a loud amplitude in general. Now, what exactly is an amplitude envelope? Amplitude envelope. So when you're recording or when you're speaking into a microphone, you can record that sound, and that can be represented as a series of amplitudes over time. Right. So on the top here, on the x-axis, I'm showing you time, and on the y-axis, I'm showing you different amplitude changes. So what the amplitude envelope is just the outline of that sound, right? So an envelope is just something that something that's the outline of something. When you put something in an envelope, you wrap it around something. When you're pushing the envelope, you're like pushing the boundaries of what's acceptable, I guess. So envelope is just the outline or the boundaries of whatever information you're talking about. So amplitude envelope is the boundaries of the sound, and so what that gives us is some information about how quickly sounds are changing, right? So if you have something like that, that's a pretty loud, rapid amplitude envelope. Right? So that's a quick changing set of amplitudes, as opposed to, I don't know, like a gong or something that's like a slow sound that has a slow amplitude envelope. And so the changes of that amplitude envelope, that's amplitude modulation. And so speech certainly requires a lot of amplitude modulation, right? Because after all I'm doing right now is modulating the amplitude of energy in the airways in front of me, and then and somehow your ears, and specifically your lateral lemniscus, is detecting those amplitude modulations and then translating that into the speech, that lexicon that's already in your heads. And so similarly, you can have frequency modulations. So in addition to changing the amplitude of my voice, I'm also changing the frequency of my voice, and you can detect that in your auditory system as well. So finally, the mediogeniculate body. I know there's a lot of Latinate terms for this, but in general, the organization of the auditory system just has more subcortical regions than there are in vision or in olfaction. So that's kind of the way the system works. But but after the level of the primary auditory cortex, there's sort of less to worry about in the auditory system. So the last part of the last subcortical way station of the auditory system is the mediogeniculate body, and that's within the thalamus. And the main role of that is just to relay information to the level of the auditory cortex. It's quite important to know, for instance, that even if you don't have a cortex, right? Even if you're a zombie and you're brain dead, right? So there's no input at the level of the cortex. Just the intact connection towards the thalamus, specifically towards the mediogeniculate nucleus, is enough to cause you to do some kinds of behavior, such as whining, right? You could, for instance, be in a coma, be in a vegetative state, and not have any input at the level of the cortex, and still orient or still turn your head when you get sounds up to the level of the MGM. Any questions? No. Okay, so I think I can get through a little bit more on the auditory system. So now we're at the level of the auditory cortex. And I just wanted to highlight a couple of auditory cortex functions, such as pitch and auditory scene analysis or streaming. So here's the brain. Right? Auditory cortex is just below the sylvian fissure. Right? So when we talked about anatomy, we talked about the sulci and gyri. Right? Sulci are inside inward folds of the brain. So sylvian fissures are kind of the most prominent inward fold of the brain, and so and that's just the dividing line between the temporal lobe and the frontal lobe. Right? So you have your Sylvian Fisher. Just underneath that are Brodmann areas 41 and 42, and that's the seats of the primary auditory cortex. So sounds generally primarily activate that area, and then structurally, there's a bunch of names that people that we use to describe that general area, and that includes the Heschel's gyrus, which is generally this area, and then planum temporal, which well, I won't ask you to differentiate between Heschel's gyrus and planum temporal, and superior temporal gyrus and superior temporal sulcus. They're all kind of in the same general area, but in general, they're all within the superior part of the temporal lobe. And so the organization, a little bit more about how the auditory cortex is organized, is somewhat analogous between monkeys and humans, but what you can see in all cases are this A1, which is again immediately below the Sylvian fissure. That's the seat of the primary auditory cortex, and then beyond the level of the primary auditory cortex, the auditory system takes a kind of core and belt and parabelt-like or concentric organization of auditory structure, such that after you get past the level of the primary auditory cortex, you have a few different regions that are kind of concentrically oriented. And so the core of that is known as the core, and then beyond that, people talk about the belt and the parabelt regions, and they are kind of the concentrically organized regions that are important for primary processing in the core, and then more secondary kinds of processing in the belt and the parabelt areas. And what's really kind of a topic of research for a while is how these regions connect to other parts of the brain, such as the frontal lobe, to enable understanding of speech and music. And so it's also worth noting that there are some asymmetries. So by that I mean that the left and the right brains are not necessarily just mirror images of each other. So most people actually have a slightly larger planum temporal or Heschel's gyrus in their left hemisphere compared to their right hemisphere. And so you might wonder why that is. And one of several hypotheses, probably the most prominent hypothesis, is that because speech is left lateralized, our left hemispheres have kind of evolved such that they've become more kind of specialized for sounds that are important for speech, right? And sounds that are important for speech especially include temporal modulations, right? So when I'm speaking, I'm modulating my sounds in a very rapidly over time. So those are temporal modulations of sounds, and so that's what the left auditory cortex is very good at. So what's the right temporal auditory cortex good at, in contrast, is frequency modulation. So different sounds that are in different frequencies or different parts of the spectrum, so that's spectral modulations. So when I'm playing music, for instance, I'm making different pitches over time. Right? So I'm making high pitches and low pitches, and those pitch differences or frequency differences are especially sensitive in the right hemisphere. So there's this trade-off, right, between spectral processing in the right hemisphere and or right planum temporal, and temporal processing in the left planum temporal. That might be kind of the functional analog or functional correlate of what gets structurally shown to be true in this leftward asymmetry of the Heschel's gyrus. So another really important ability or characteristic of the auditory system is that we're good at understanding crowded environments of sounds. And so this is known as the famous cocktail party problem. I don't know why it's called cocktail party. It should be called pizza party or just any kind of crowded situation. And so the cocktail party problem is it's just a great demonstration of how we can use selective attention with our auditory systems in order to make sense of the world, right? So imagine if you're in a crowded cocktail party or pizza party or mingling with a lot of people, you are actually, for most people, there's actually very little difficulty to hear one person's voice versus another person's voice. Right? So all I have to do, if I'm maintaining one conversation with another person, all I have to do is like switch my attention in order to eavesdrop on another conversation that's going on in a different part of the room. Right? So that's a pretty remarkable ability of the auditory system, and like no computers can do this, right? They have not been able to engineer computers that can switch attention like that. And then the other important characteristic is that if I'm continuing my conversation with you in this crowded cocktail party situation, and then somebody mentions my name somewhere else in the room, like that automatically drives my attention towards that area, right? So there's this breakthrough of the unintended into my conscious awareness. That's again part of the ability to switch attention very flexibly. And so that's all part of the cocktail party problem, which kind of tries to answer the overarching question of how the brain can hone in on a single speaker when in a crowded or noisy environment. And that's not very well solved. But we do know that one of the strategies or techniques that our system uses to solve this cocktail party problem is auditory stream segregation. And so that's just the ability to separate different parts of the environment based on different features of sound. So one feature is pitch, another feature is timbre, another feature is temporal coherence. And so I'll just give you an example of what that sounds like. I'm going to play, actually there's a little introduction that introduces what the sounds are that we're going to play. But it's basically the same tune that is embedded in another tune. And so what they're going to do is they're going to start by playing the target tune interleaved with the non-target or the distractor tune. And then they're going to do it again, but this time there's going to be more of a pitch separation, such that the target tune is in one region of pitch and the other tune is the distractor tune is in a different region. And they're going to keep doing it until at the end you should be able to identify the familiar tune in the whole clip. Okay.
Demonstration 5. Segregation of a melody from interfering tones. A familiar melody is alternated with distracting tones that are drawn randomly from the same pitch range. On each presentation, the melody is transposed upward in pitch. Note the point at which you can identify the melody.
Okay, did anyone hear the melody towards the end? What's the melody? Yeah, we have a little lamb. Right. So, yeah, it starts out completely unrecognizable because it's interleaved with the distractor melody that's in the same range, right? But then over time, you can hear that they start to tease apart in pitch the familiar melody versus distractor melody, and that's how you can start to separate that percept into two separate streams, right? So that's auditory stream segregation, is that you're segregating your sounds into separate streams, and when you're hearing them as separate streams, you can start to recognize the familiar tune as opposed to the distractor tune. It's pretty cool. It's too bad that there's this rattling going on. I don't know why that's there, but okay. Any questions about that? Okay. So I'll spend a little bit more time on higher order auditory functions, which are speech and music. So so after the level of the auditory cortex, the system, the whole brain includes a whole set of different regions in order to comprehend and to produce speech and music. Right? So a really nice example comes from this ECOG study that I've already shown you, I think, when we talked about ECOG in general. And so this is, of course, a very high temporal and spatial resolution because we're looking at open brain recordings obtained from patients doing open brain surgery for epilepsy. And so what they can do is like stack a grid of electrodes on the side of the cortex. And so from that you can get coverage of the lateral parts of the superior frontal gyrus and also of the lateral frontal lobe. And so what this is very elegantly showing, this is a classic verb generation task. Right? So people are given a stimulus, which is a noun, and the subject task is to come up with, is to think of a verb and to say that verb out loud. Right? So when I say ball, road, and if I say pen, you might say right. And so this, you could probably be able to do this in like well under 1 second. Like it's a very easy task. It takes them about 154 seconds, but these are open brain surgery patients, so keep that in mind. So it's pretty remarkable that they were getting people to do this during open brain surgery. So what they're doing right now is showing the stimulus right at 0 milliseconds, and then immediately you can see this whole temporal evolution of cascade of events that are going on throughout the body. So first there's this auditory related activity, right, that's in the superior frontal gyrus, and then that then goes on and passes forward towards the frontal lobe activity, specifically anterior portion of the frontal lobe activity. So this is really middle frontal gyrus, which then goes on towards the inferior frontal gyrus, which is the classic Broca's area, and then afterwards you get the motor area. So it's as if you are getting your auditory system to be active, which then gets your thinking parts of your language system to be active, and then which then gets your motor execution system to be active. Right? So there's this whole cascade of events that happens that goes from auditory to cognitive to motor. And so that's just a really kind of neat way to pull together all these different areas of the brain that are at work beyond the level of the auditory cortex, but that are activated by the auditory system. So when you're talking about speech, a really important part of the stimulus comes from the voice itself, right? And so one question you might ask is, how do we manage to hear our own voices? And a neat idea that I'm going to present to you now that will come back in future lectures is this idea of a difference copy, also known as a correlated discharge, which is just the idea that our brains can tag our own actions as our own, right? So one example is that when you're listening to your own voice, you know that that is your own voice, and that causes you to your auditory system to react to your own voice a little bit differently compared to if you're just listening to a different person speak, right? So how many of you have like listened to yourself recorded? Right? How many of you think that sounds kind of weird? Right? It's very strange, right? So hearing your voice played back to you sounds categorically different and qualitatively different compared to hearing your own voice as you're saying it. And so that should give you a clue that that we have this thing known as difference copy, which is our brain's ability to know that our own actions are our own actions and thus not over-respond to it. So in fact, one of the many hypotheses underlying schizophrenia, right, which often comes with hearing your own voices, is that there's a broken efference copy. Right? In other words, the brain is unable to detect one's own actions or sounds or voices as one's own. So how does the auditory cortex do this? Firstly, it can suppress itself during speech. Right? So this is a very simple example of how you can suppress your own auditory cortex. This is an ECOG study, and again, you're looking at the right hemisphere here. Each black dot is a recording site, which is an electrode that is recording from brain activity at one time. And so in red, I'm showing you the power or level of activity that is induced by hearing sounds, whereas in blue, I'm showing the activity when you're hearing your own voice, and so or producing your own voice. And so when you're speaking, you're suppressing. So the blue is lower than the red, right? So you're suppressing your own voice or neuroactivity due to your own voice when you're speaking. And so another really neat way to demonstrate this is using this technique known as perturbed auditory feedback. And so how that works is you're being put in a room, you are listening through headphones, and you are given a mic. It's like a karaoke situation, right? So you have a mic, you're talking into your headphones. And you give people a tone, like that, and your job is just to hum it back, right? But what we do when you hum back the tone that you get, the target tone that you're supposed to hum back, is we can record the sound and then immediately we can play it back, but we can transpose up or down in frequency what you're hearing back. So essentially, we're tricking you into thinking that you're singing out of tune. And so what most people do when they're confronted with this perturbed auditory feedback situation is they compensate by singing in the opposite direction of the perturbation. Right? So if I tell you, if you're supposed to sing an A, and I make you think that you're singing an A sharp, you would sing flat in response. Right? So that's it's a very kind of natural and almost involuntary response. So we can record how much you're deviating from the perturbation and how much you're deviating from the target. Right? And based on that, we can get the sense of how perturbable your auditory system is. And based on that, you can get some inference for how strong your efference copy is. And so this is an MEG study, which I think is the first in its kind. What they're doing is recording magnetoencephalography from human brains while people are listening to their own voices or listening to transposed or altered versions of their own voices or listening to other voices, right? So we call alien voices. And what you're generally seeing is that when you're hearing the pitch-shifted version of your own voice, you get an attenuation or a decrease in activity as a result of the pitch shift. So, however, when you're hearing somebody else's voice with the alien voice, you get a release from that attenuation. So, any questions about that? Yeah.
In the previous slide, you did something about how you could see how strong your efference copy is based off of that.
Mm-hmm.
So the more you would like alter yourself in relation to the sound,
yeah.
the more you would like change and like try and bring it up or down or whatever.
Yeah.
Would that like show greater efference copy or lower?
Yeah, so a stronger efference copy would probably mean that you're more perturbable. So you would adjust more. But I think that's a really good point because there's only a certain range through which it works, right? Because if I perturb the perturbation so much that you de-identify with it, then you won't really shift at all, right? So and that's kind of what you can see here is that the pitch-shifted auditory feedback results in people. It results in activity that's closer to the alien situation. Right? So that just means that when I trick you into thinking that you're a completely different speaker, you're not going to be tricked because that's so far from what you've actually identified as yourself that you stop identifying with the perturbation. So that's a really good point. But in this situation, what would really work is this situation, what works best is like a small but noticeable amount of perturbation. Yeah, so there's it's a very interesting paradigm. Okay, so at the last 10 minutes or so, I'm just going to talk a little bit about this really fun experiment, which gets into what much more about what I do, which is about music and brain. And so in this very nice paper, they looked at how we derive pleasure from music. Right? And so they played people, so the researchers in Montreal a couple of years ago, I think it's like 4 years ago when this came out, what they did was they went on iTunes, you've all been on iTunes, right? And they downloaded all these different clips of music that you're unlikely to have heard. So they're like kind of relatively unknown pieces of music. They're not like the top 10 or whatever. And they just played them, and they asked people to do this bidding paradigm, right? So say, okay, here's a bunch of money, and you can use this money, and you can listen to the music, and you can either not buy it, so pay $0 for it, which would mean you don't like it, or you can pay 99 cents or $1.29 or $2 for the piece of music, okay? And so you actually get to keep this music. All right, so I'll just play you a couple examples. Okay, say if I gave you $10, how many of you would spend $2 on that? Okay. How many of you would spend like $1.29 on it? How many people would spend 99 cents on it? How many would spend no money, none, just keep it? Okay, great. All right, so what about this example? Okay, starting with $10, who's going to spend $2 on that? Nobody? Okay, right. $1.29? 99 cents? Wouldn't buy it at all. Oh, you could like me. Okay, okay, great. So I mean, but the point is, like, sometimes you'll get people who will only buy the second piece, right? And so there are different, individual differences in preferences. But the point is that for whatever you like to listen to, you get these changes in your parasympathetic nervous system. You get heart rate changes, you get skin conductance changes, and you get breathing rate changes. And what Salimport et al. Did, is a very nice paradigm, was pick this music buying paradigm and have people listen to these excerpts of music and decide how much they would bid on these pieces of music. And then they recorded fMRI while they were listening to these desirable and undesirable pieces of music for each person. And what they found was that the level, there was more activity in the reward system for and we won't talk about specific regions of the reward system just yet, because that's for much later in the course, but just suffice it to say that they found more activity in the reward system for pieces of music that were purchased compared to pieces of music that were not purchased. Right? And furthermore, what they found was that the auditory areas, so in the superior temporal gyrus, was more functionally correlated with these reward areas during the perception of music that people were willing to purchase. So the more you like the piece of music, the more you're actually getting synchronous or functionally connected or correlated activity between your auditory system and your reward system. So that really tells us that the auditory system doesn't really work on its own. It does work with a bunch of other regions, including the reward system. And that's what Sean's thesis is about. Okay. Any questions? Okay, so it's only 2:34, but I think that's all I had. I'm going to actually make sure I save this. And so a few times ago, I've managed to